Welcome viewers. Today I will be talking on B vitamins. 8 water-soluble compounds are universally accepted as the B vitamins. In my previous lecture on this series, I talked about the first 4 of the B vitamins, vitamin B1, B2, B3 and B5. Today, in this lecture, I will be talking on the remaining 4 of the B vitamins, vitamin B6 or pyridoxine, vitamin B7 or biotin, vitamin B9 or folic acid, and vitamin B12 or cobalamin. It is important to note that all the B vitamins function as coenzymes or cofactors of several enzymes that are involved in the intermediary metabolism, or in other words, B vitamins regulate the internal biochemistry of the cell. We start with vitamin B6. Vitamin B6 or pyridoxine has a molecular formula of C8H11NO3 and a molecular weight of 169. Food rich in vitamin B6 include legumes, nuts, eggs, meats, breads, and several different cereals. The recommended dietary allowance is 1.3 to 1.7 milligram for males between age 19 to 70 years. And the dietary deficiency syndrome associated with this vitamin is anemia and peripheral neuropathy. There are several forms of vitamin B6 that are there in human body. They are pyridoxine, pyridoxine 5' phosphate, pyridoxal, pyridoxal 5' phosphate, pyridoxamine, pyridoxamine 5' phosphate, and the pyridoxic acid, which is the catabolite form excreted in the urine. However, of these 7 forms, pyridoxal 5' phosphate or PLP is the most metabolically important form. Now, the functions of vitamin B6 include transamination and decarboxylation reactions. Pyridoxal phosphate is mostly associated with the enzymes that are responsible for catabolism of amino acids. And pyridoxal phosphate is involved in synthesis of neurotransmitters, synthesis of histamines, it's an immunomolecule, and hemoglobin, the molecule that is responsible for transport of oxygen from lung to different cells and tissues. PLP is involved in synthesis of biologically active amines from decarboxylation of amino acids. The notable examples include histamine, that is involved in inflammation reactions, and it is produced from decarboxylation of histidine, production of serotonin from tryptophan, and production of GABA or gamma aminobutyric acid, which is an inhibitory neurotransmitter, from glutamate, and synthesis of dopamine from dihydroxyphenylalanine. Dopamine is the molecule that is synthesized in the brain, and we know that in the case of disease, dopamine synthesis is severely impaired. This slide shows one of the important transamination reaction that is catalyzed by pyridoxal phosphate. Here, the amino acid aspartate is transaminated in presence of aspartate transaminase to form oxaloacetate, and the amino group is removed from aspartate and is taken up by alpha ketoglutarate, and glutamate is formed. Similarly, by the action of alanine transaminase, pyruvate is produced, And by the same fashion, the amino group is accepted by alpha ketoglutarate to form glutamate. This reaction shows synthesis of GABA or gamma aminobutyric acid, the inhibitory neurotransmitter. It's a decarboxylation reaction and catalyzed by the enzyme glutamate decarboxylase, which requires PLP, and a molecule of carbon dioxide is released, and glutamate gets converted to gamma aminobutyric acid or GABA, an inhibitory neurotransmitter. As I told you that vitamin B6 deficiency leads to anemia. This is because inside our body, glycine and succinyl-CoA gets combined to produce ALA or gamma amino levulinic acid, and the enzyme responsible for this reaction is called ALA synthase, which requires PLP for its function. And ALA or amino levulinic acid is directly responsible for the synthesis of heme moiety, the important part of hemoglobin. This cartoon summarizes all the reactions that are aided by PLP. On top are the decarboxylation reactions, where we see tryptophan is decarboxylated to form serotonin, tyrosine to norepinephrine, and histidine to histamine. This reaction over here shows the synthesis of ALA by combination of glycine and succinyl-CoA to form ALA, which leads to the synthesis of heme. The reaction over here is the reaction of glycogenolysis, where glycogen is broken down to maintain blood glucose level, and the enzyme that is responsible is glycogen phosphorylase, which requires PLP for its function. And these three reactions over here are the ones that are transamination reaction, where alpha ketoglutarate accepts an amino group to form glutamate, alanine is transaminated to produce pyruvate, and aspartate is transaminated by aspartate transaminase to produce oxaloacetate. It is important to note that there are about 140 enzymes that require PLP as a cofactor, and this accounts for almost 4% of all the enzymes that are acting inside the cell. This picture shows a co-crystal of glycogen phosphorylase along with the cofactor PLP. This enzyme is responsible for breaking down of glycogen to produce glucose through the process of glycogenolysis. B7 is biotin. The chemical formula is C10H16N2O3S, and the molecular weight is 244. Foodstuff rich in vitamin B7 include nuts, fish, egg, milk, and different vegetables. And the recommended dietary allowance is 30 microgram for males between age of 19 to 70 years, and the deficiency syndrome associated with biotin is dermatitis and enteritis. The biochemical role of vitamin B7 or biotin is its involvement as cofactor with different carboxylases. These group of enzymes are involved in transport of word carbon molecule in the form mostly as carbon dioxide, and these enzymes are responsible in fatty acid biosynthesis, gluconeogenesis, and synthesis of amino acids like valine and isoleucine. Fatty acids are essential for biological membranes and absorption and transport of fat-soluble vitamins. gluconeogenesis is an important metabolic process because human brain very much utilizes glucose, and this mechanism is there to ensure steady glucose level in the blood to be used by the brain. In case of severe deficiency of carbohydrate in the diet, gluconeogenesis is the pathway through which blood glucose level is maintained and glucose is synthesized from non-carbohydrate sources like proteins and fats. And amino acids like isoleucine and valine are required for synthesis of proteins. There are 9 enzymes that require biotin as the cofactor, of which 4 are present and functional in humans. The very important one is pyruvate carboxylase, which is responsible for formation of oxaloacetate from pyruvate. This step is very much important both to run TCA cycle, the major step for energy production inside the cell, as well as the reverse reaction is important for gluconeogenesis, where pyruvate is formed and from which glucose can be prepared. Another important enzyme that requires biotin as cofactor is acetyl-CoA carboxylase, the enzyme that is responsible for formation of malonyl-CoA from acetyl-CoA. And formation of malonyl-CoA from acetyl-CoA is the first and important step in fatty acid biosynthesis. We will see. This cartoon shows the reaction of pyruvate carboxylase, where pyruvate is used by a carboxybiotin cofactor, and the carbon dioxide is eventually transferred onto pyruvate, and pyruvate is converted to oxaloacetate and carboxybiotin becomes biotin. This is the step catalyzed by acetyl-CoA carboxylase, and acetyl-CoA carboxylase acts on acetyl-CoA to form malonyl-CoA, and this reaction also requires ATP and enzyme biotin complex for functioning. Vitamin B9 or folic acid, molecular formula C19H19N7O6 and a molecular weight of 441. Food rich in vitamin B9 include dark green leafy vegetables, pork, shellfish, liver, bread, and so on. And the recommended dietary allowance is 400 microgram for males of age 19 to 70 years. And the dietary deficiency associated is metabolic anemia, and it is important to note that if a mother is deficient in folic acid while pregnant, then the newborn baby may develop severe defects in brain and spinal cord. That's why in all developing countries of the world, pregnant women are always given folate as a dietary supplement. Folic acid got its name from the Latin word folium, which means leaf. That's why folic acid is rich in green leafy vegetables. Normally, it takes about months for folic acid deficiency to develop because folic acid remains stored up to 20 milligram in human body. Thus, when all this store is depleted, can only the deficiency symptoms crop up. Folate is required for rapid cell growth and division. That's why it is very much important to have folic acid during infancy and pregnancy. These days, it has been observed that folic acid can prevent symptoms of arsenic toxicity. As we know that in Gangetic West Bengal and in Bangladesh, there is a severe problem of groundwater arsenic contamination. And people who are affected with arsenic toxicity, they develop skin lesions like keratosis, skin warts, difference in pigmentation, as well as skin cancer. And it has been observed by many researchers that folate supplement in their diet prevent manifestation of these skin lesions upon arsenic toxicity. Folate coenzymes are involved in different biological reactions like synthesis of DNA. We will see that pyrimidine nucleotide like DTMP is formed from DUMP, and folate is an important cofactor in this reaction. In purine synthesis, formation of glycinamide and 5-amino-4-imidazole carboxamide requires folate. So this is how folate aids in DNA synthesis and repair. Folate is also very important to maintain the formate pool and utilization of formate in body. An interconversion of amino acid, for example glycine to serine, catabolism of glutamate and metabolism of homocysteine is governed by the coenzyme folate. Here we show that vitamin B9 is involved in the interconversion of amino acid glycine to serine. This is performed by serine hydroxymethyl transferase, and the enzyme requires folate. This cartoon summarizes all the reactions that requires folate as a coenzyme. For example, synthesis of DTMP from DUMP requires folate. Synthesis of methionine from homocysteine requires folate. Synthesis of glutamate requires folate. Utilization of formate and maintaining formate pool is also folate dependent. Interconversion between glycine and serine is also folate dependent. And finally, synthesis of purine and pyrimidine nucleotides are dependent on folate. Now, as I told you that if a pregnant woman has folate deficiency in her diet, then serious consequences can happen. The baby that will be born may develop problem in spine, skull and brain. The most dreaded disease is spina bifida, where the spinal cord of the baby is not closed properly, it's open, and these are the photographs of spina bifida newborn babies. Finally, we come to vitamin B12 or cobalamin. Cobalamin has a formula of C63H89, cobalt, N14, O14 and P, and the molecular weight is 1356. Food rich in cobalamin include eggs, poultry, cheese, milk, meat, and the recommended dietary allowance is 2.4 microgram for males of age between 19 to 70 years, and the deficiency disease associated with vitamin B12 is megaloblastic anemia. Some important facts of vitamin B12. Vitamin B12 is the largest and structurally most complicated of the B vitamins. It contains a biologically rare element, cobalt. It is important to note that only eubacteria and archaebacteria can synthesize vitamin B12, and that's why industry has to rely on fermentative production using bacteria and archaebacteria. However, once the hydrocobalamin is formed by the bacteria, it can be converted into different forms of cobalamin like methylcobalamin and others inside human body. Cobalamin plays a key role in normal functioning of brain and nervous system. It is important for production of blood, and it is involved in DNA synthesis. As I showed earlier, that tetrahydrofolate is important for production of purine and pyrimidine nucleotides. Now, production and maintenance of tetrahydrofolate, on the other hand, is dependent on cobalamin, and for the same reason, cobalamin is also important in fatty acid synthesis. Now, megaloblastic anemia is a disease where immature and large-sized red blood cells come in circulation. They are not so rich in hemoglobin, and the white blood cells also show multilobulated nucleus. Megaloblastic anemia can also result from destruction of the gastric parietal cells, and once these gastric parietal cells are destroyed, the secretion of an intrinsic factor is impaired, and this impaired secretion of intrinsic factor leads to impaired absorption of vitamin B12, which causes this megaloblastic anemia. I'd like to conclude with a note that vitamin B12 is of concern for vegans because till date vitamin B12 is found only with animal products. Although there are some reports of plant products in which vitamin B12 is present, it is still controversial and not proved beyond doubt. So, vegans must take vitamin B12 supplements. We come to the take-home message of this talk. 8 vitamins are universally recognized as the B vitamins. They are vitamin B1, B2, B3, B5, B6, B7, B9 and B12. All these B vitamins are water soluble and they are responsible for the internal biochemistry of the cell. Most of the B vitamins act as precursors of coenzymes and cofactors of several enzymes. The coenzyme form of vitamin B6 is pyridoxal phosphate. Biotin or vitamin B7 itself acts as cofactors. Coenzyme form of vitamin B9 is tetrahydrofolate and vitamin B12 is cobalamin. The diseases associated with vitamin B deficiency include beriberi, pellagra, peripheral neuropathy, dermatitis, enteritis, and different forms of anemia. And vitamin B12 is of concern for vegans because it is found only in animal products. Thank you.